It will be a pleasure to introduce Matteo Degiacomi. So Matteo got his PhD at EPFL under supervision of Matteo Del Ferraro. Then he moved to Oxford for a postdoc in the Carol Robinson's lab. And then in 2017, correct, he got the first step of independence by having an EPSLC fellowship in Durham. And in 2020, became associate professor there. And I think from 1st of November, correct?
Correct.
He is actually a reader in Edinburgh. So he moved a bit northern.
Even more.
Yeah.
Even more rain.
Even more rain. So to you, Matteo.
Thank you. Great to be here today. Right, so the story I have today is about how we can use machine learning and combine it with molecular simulation to understand a little bit more about the conformational spaces occupied by proteins. So starting just with a picture that is, let's say, the traditional picture we are taught, the structure determines the function. So let me go to my slides first of all, and then I can change. There we go. So we know that an organism has a certain shape that enables us to carry out a certain function. You can think about a human body. Now, this paradigm, of course, casts down to every level of complexity, down to a simple molecule, which justifies why, as computational biophysicists, we are interested in studying the structure of molecules. So if you understand the structure of a protein, in this case, so you see the blue squiggle there, this is something that we may use to infer what that function so how this function may impact a whole organism. And in fact, that's probably very much underscored with the Nobel Prize award this year. It's very interesting for us to know the structure of proteins because that's often the first step to understand diseases and ways of curing them, for drug design, for instance. Now, of course, this is true, but it's kind of part of the story only because true, the structure determines the function, but dynamics plays a very important role as well. So what you're seeing here is a molecular dynamics simulation, a technique that we've been using also heavily in my group and even before. And well, if we truly want to understand how a protein operates in an organism, this is the kind of picture we want to get. We want to know what's the whole ensemble of different conformations the protein takes. So this is essentially an overlay of more and less probable states. And this is all the more important considering that proteins very often operate as part of a complex. So about 70-ish% of proteins actually carry out the biological task as part of a complex. So to understand what a protein does, we may have to understand how it occupies this conformational space, and then how through its dynamics it can bind to its binding partners, may them be small molecules or may them be other proteins. And through the phenomenon of emergence, we may end up with a complex that can carry out a task more complicated than the task that an individual protein may be able to carry out. So the issue is that now getting this kind of information experimentally is tricky. Getting information on all the states the protein occupies is really, really hard. So typically we have X-ray crystallography, NMR, electron microscopy that give us some insight on the structure. But definitely rare events, which are very often the things we care about, are the ones that lay outside of what we can gain structural atomistic insight into. And studying the structure of protein complexes is even harder than studying the structure of individual molecules. So that's where we're at. And what I would like to do now is to start with about 4 minutes of a kind of historical perspective of my own work to create a pretext to justify then why I'm interested in my group in the usage of machine learning to gain insight into protein structure. Right, so historically, I would say, one thing we would do to understand how a protein assembles into a complex, since this is something that is maybe tricky to do experimentally, we may go through a process called protein docking. Many of you are probably familiar with that. Essentially, the idea is the following. If you have information on the building blocks of a complex, here represented by Lego bricks, so different proteins, what we're trying to do is how can we stick together all of the different proteins in the complex that has a biological significance. Now, if you ever played Lego, you know that there's one way to follow the instructions and getting the right model, but there's many different things you can do with your pieces. Well, now, and so of course, some of these complexes do look like a complex as well, but some of them mean nothing at all. It's just a pile of bricks. So the question is, how do we try to manipulate all our building blocks to select among all the many, many different combinations, the correct one? So one thing we can do is to actually guide our modeling with some extra information. So typically we have a physical model that tells us about interatomic interactions. So we want typically to generate the model of lowest energy. And if we had a way to calculate with incredible precision the energy of a single complex, and we had a really good optimization algorithm able to explore the conformational space, explore all those different complexes, we should in principle be able to find the complex of lowest energy. Problem is that we don't have that. So if we add some extra information, typically experimental data, we can help our algorithm weeding out what among the many models, besides having what our approximate function for the energy tells us it's a good model, well, weeding out which one actually is also consistent with experiments. And so this is what we have been doing historically. So we developed even an optimization engine for that called POW. And this is now an idealized picture I'm giving you. I'm showing you Lego bricks and I'm telling you how you can assemble them into complexes. But just in the previous slide, I've told you that proteins are wobbly. So they are not really Lego bricks. So the question is, what can we do to bring this idea of flexibility? And for that, I'll take another historical work we've done. And I'll quickly tell you about a protein that was both my favorite and my most hated protein because I did a PhD on that. And I'm sure many of you will be familiar with that. So this is called aerolysine. It's a pore-forming toxin. We know the atomic structure of the monomer. You can see it up there to the top left. And pore-forming toxins are ubiquitous in all kingdoms of life. And what they do, they drill holes into the membranes of the target cell. And the way to do that for them is to bind onto the target cell, onto the surface of it. Then they multimerize. And after the multimerization is complete, we have a massive conformational change that leads to the insertion of a barrel. So in this case, aerolysine forms beta barrel. And this is what kills the cell. So you have either osmotic shock or inflow of calcium ions, and this is what leads to apoptosis. Now, what you're seeing here is the models we produced at the time. So you can imagine at the time we had only the electron density of the complexes of both what we call the pre-pore, this kind of volcano conformation, and the pore states, so this disk. And we were asked by our biologist collaborators how we can build atomic models based on the atomic structure of the monomer. And at this point, you can probably see yourself, it's quite hard to imagine how you can build both a volcano and a disk just with a protein treated like a Lego brick. You can see now the overlay here of models we produce with the electron density. And the reason why we could do that is because we actually run molecular dynamics simulation of aerolysine to gain some insight on how it moves. And we try to couple this information with protein docking. And what we did is something that, of course, many people before us did. When you have a molecular dynamics simulation, you can run a principal components analysis. So essentially, you're creating a representation of your data. This is where we start using a bit more machine learning language. So we have the protein is a high dimensional data point. The degrees of freedom of your system would be the X, Y, and Z coordinates of your atoms. So if you have n atoms, you have 3 times n degrees of freedom. What we're trying to do is to create a low dimensional representation that captures the whole conformational space occupied by the protein in simulation. And what you're seeing in this plot here, that's a projection into a 2-dimensional representation. This is a 2-dimensional eigenspace where every point is a snapshot from the simulation. And the interesting thing about having this kind of representation is now we have some information on the adjacency between different conformations explored in the simulation. So what we did is combine the classical idea of protein docking, so taking our subunits, moving them in space, but now which subunits? Well, what we would do, we would actually pick a coordinate in this eigenspace, select a coordinate, locate the closest snapshot from the simulation, and use that as a building block for the assembly of our model. Now, by combining rotor translation and selection of conformants from our simulation, give it to POW, you get the models you've seen in the previous slide. Now, the question is, why am I telling you this? And the reason why I'm telling you this is because what you have here is a representation of the data. So let's zoom in. But so principal components analysis is good at recapitulating data. But what I really would have liked at that time is a way to say, well, this is how the data is distributed. But I would like to be able to generate more data beyond what has been explicitly sampled in the simulation. So what is that, essentially? And in principle, this is something you can do with the principal components analysis. You can just take every dimension is represented by one eigenvector. You just take a weighted sum of your eigenvectors, and you get something that kind of looks like a protein out. But usually, the structures you get on the other sides are quite outrageous. So they vaguely look like a protein, but there's heavy deformations. So we wanted to have an atomistic model, something that is as accurate as possible, and PCA did not cut it. So we started thinking, is there a different way we can create representation of my data that I can more effectively exploit to generate conformations that I can then use, in this case for protein docking, and maybe, as we'll see later, for other tasks as well. And this is where we started looking into machine learning. And in principle, principal components analysis is an example of machine learning, but there's plenty more examples. And if we look in the direction of neural network, one of the most traditional of them all is the autoencoder. So essentially, what is an autoencoder trying to do? Well, you have some high-dimensional data, that the variable x on the left-hand side. What you're trying to do is to compress the data and then decompress it so that the loss of information is minimal as the data travels through. So what you want to have essentially is that in an ideal world, the data you get on the other side is identical to the data you put in. That means you can compress and decompress the data with minimal loss. And typically, this loss is quantified by a mean square error between input and output. So the question is, well, can we just say that X is a protein conformation? And I'm hoping to come up with a way of compressing the data or encode the data and then decode the data, so decompress it, so that what I get on the other side is still a protein. So if we can do that, what we can imagine, I can take a molecular dynamics trajectory. And when I train my machine learning model by compressing and decompressing the data, what I'm obtaining is actually a way of producing a low-dimensional representation. This is what's called the latent space. And this low-dimensional representation, well, that's a place where every single conformation in my simulation projects into. But now if we look at this decoder, so this decoder is a neural network that essentially is a little machine that produces data that is as similar as possible to what is being trained with. So it's a kind of, hopefully, a little machine that produces protein conformations by just putting, in this case, just two numbers in on one side. And if we can do that, well, we can actually pick coordinates elsewhere in this graph and generate conformations that do not exist in the data set, but hopefully they are consistent with everything the neural network has seen. So that's the idea. And so back in 2019, which in machine learning is basically a lifetime ago, we tried this. And we tried this on this protein here, the P24. It's part of the HIV-1 capsular. So in this case, we have a simulation of the monomer. And well, classical thing you do, take a simulation, shuffle it, separate it into parts for training and validation, use the training data set to train the network, and then check whether you can reproduce the validation structures. And we were quite happy with that, because in fact, now if we look at this graph here, I'm reporting in this graph the root mean square deviation between the known structures in the simulation and the ones the neural network would produce. And we had a root mean square deviation under 1.5, which for protein structures is pretty good. And since it all started with the idea of protein docking, the question is, well, if we have a little machine that generates conformers, can we use this to generate conformers that are useful for protein docking? And that justifies actually why we thought about using the P24 protein. It's part of the HIV-1 capsular, so it's actually a protein that operates as part of a complex. And we so happen to know already the structure of a complex, so this becomes an interesting benchmark case. So what we did, well, take the simulation, train our model, as I described before, and then give this model to POW, so the docking algorithm. And now what POW was asked to is to actually to ask the neural network to generate conformers and use them for docking. And what we got is this overlay, is the model in red, which is now overlaid with the gray structure, the known complex. And what was quite exciting at the time was that actually the conformational changes suspected for the protein as it switches from the unbound to the bound state, or the root mean square deviation between the known atomic structure of the unbound state and the known atomic structure of the bound state, is quite large, 10.4 amps. So there's two domains that need to be arranged, one with respect of each other. In the simulation, we find conformation, the simulation of the unbound state, we find conformations that are actually much closer to the bound state, 3.2 angstroms. And the thing I found quite exciting is that if we now ask the question, what's the root mean square deviation between one of the subunits here in the complex that's been used to build the complex and the non-bound state? Well, the root mean square deviation here was 2.7. So the neural network could produce a conformer closer to the bound state than anything it was provided with, and the docking algorithm could fish it out. And so this was quite exciting. And however, when we started really looking very carefully at the models generated by this quite naive neural network, classical feed-forward perceptron, we noticed that actually the devil is in the details. And more specifically, so if we truly looked at the atomic structures very carefully, still better than a principal components analysis, but still they were not physically accurate. Bonds were stretched, angles were a bit too open. Let's not talk about their Ramachandran distribution. So they looked better than what you can do with a PCA, but still they were not real atomic structures of protons. Then it was actually quite of a pain to train the neural network. And the reason is because we were essentially treating every degree of freedom of the protein with a neuron. So the first layer had a number of neurons commensurate to the degrees of freedom, same on the opposite side. So this was associated with many millions parameters. And it so turned out that actually we needed to run many repeats to get to models that performed as well as the one I've shown you before. And even more annoyingly, since we're dealing with an architecture that in the first layer needs to have a neuron per degree of freedom, what that means is that every time I train the model on a new protein, I need to change the architecture of the model, which is annoying. And so we started thinking, can we address those problems? And this has led to the collaboration with Chris Wilcox in the Department of Computer Science in Durham, and who's worked with Venkat, my postdoc, and Sam, PhD student, who actually graduated 2 days ago with much of the work I'm presenting today. This makes me very happy. So the idea we came up with to actually address the problems in the previous slide is, well, actually, there's something more we know about the systems we want to learn from, about the data we want to learn from. We know physics. We know that whatever we generate should respect the laws of physics. And we know topology. So we know that atoms have relationships with each other. There's a covalent bond network that links all those atoms. And the question is, can we use this information to improve the quality of our models? So now here at the bottom schematically, you see the simplified representation of an autoencoder, what I described a couple of slides ago. Now, to bring in this idea of physics, we come up with a different way of training the model. So we slightly complicated it to encourage the model to better generalize. And the way we did it is by complicating the architecture this way. And this schematic is telling you this thing. So we don't train the neural network showing one example at a time, but we show pairs of randomly selected structures. Those pairs of structures, each of them will project somewhere into this latent space, this low-dimensional representation. And of course, we want the neural network to be able to decode those structures so that the root square deviation between input and output is minimized. Of course, we want that. But the other thing we do, we regularize the network by saying that actually, if we pick any random intermediate between those points, we want to generate a structure that has an energy as low as possible, an energy assessed with a molecular dynamics force field. So we use the amber force field here, Fi14Sb. So these actually help regularizing the network, and I'll show you later, it does lead to nice results. And for the topology, this sounds like a geeky detail, but I want to share it with you because I like the idea. So we realized that we can actually process the data not with a classical neural network, but instead with a convolutional neural network. So I want to quickly just share with you what we mean with convolutions here, and what implications of that are, as we'll see, quite useful for us. So the question is, right, convolutional neural network, how do you do convolutions on a point cloud? Protein is essentially a point cloud in the 3D space. So convolutions are typically used when processing images. So if the idea, we try to draw an analogy between the fact that the protein molecular dynamics simulation is a sort of movie of the protein, and the fact that, well, a movie is a series of images. Now, convolutions are mathematical operations that rely on the idea that the data you have the data points, the coordinates of your data set have a topological relationship. In an image, the pixels have a certain adjacency information. I cannot just take the value of 2 pixels and swap them. I don't have the same image anymore. Now, the same idea of I cannot just swap 2 atoms and hope I have the same thing also holds for molecules. So I cannot just take 2 atoms and swap them. But a protein is not an image. So where's the analogy here? How can we do convolutions on a point cloud? Well, what we thought is the following. If we imagine the points the atoms of a protein as points in space, the topology of the protein tells us that we can essentially order them. And this is something that for many of us is probably not surprising. When we go in the protein data bank and we download the structure of a protein, we know that when you've got atoms in our protein data PDB file, if 2 atoms are adjacent in this list, probably they are also adjacent in space. And probably they are also physically connected either directly with a covalent bond or indirectly through an angle or a dihedral. And this is where the idea came to us. We thought, well, how about we take the 3-dimensional coordinates of our proteins, and now each atom is described by an X and a Y and a Z coordinate. So when we have images, what we do, we treat the red, the green, and blue channels, and the red and blue colors as separate channels for our machine learning models. Here, the X, the Y, and the Z can be our channels. So what we can do 1-dimensional convolutions by sliding down the X and Y and the Z coordinates. And this is how we actually built a model that is pretty simple. But the nice feature of convolutions is that they don't care on the dimensionality of the data. If you have a bigger image, you just convolve more. So in this case, if you have 1,000 or 2,000 atoms, also it doesn't matter. You keep convolving until you get to the end of the data, which means that using convolutions, we can deal with data of arbitrary size. So we don't need to redesign the architecture of a neural network for every protein we want to deal with. So does it work? So for that, I would like to tell you about a test case we used that's called MERD. That's one of those proteins in the protein data bank that has been solved atomistically in 2 different states. So that's a protein used by bacteria for sphingolipid biosynthesis. So it's a drug target. If you prevent the bacteria from synthesizing sphingolipids, the bacterium dies. And so the 2 crystals we have, the blue one is the closed state, it's bound to a substrate. Open state is able, so by itself. So what we did, the first thing we noticed is that actually the relationship between the open and the closed state can be coarsely described by a reorganization of the little spherical domain at the top with respect to the long domain at the bottom. So literally, we have kind of some rigid body motion. So we run molecular dynamic simulation of both states, and we trained on neural network. And what you're seeing here, and I'll spend a minute on this image, is a representation of the latent space, so of the internal model the machine learning, the neural network produced. So first of all, in this case, I'm showing you a 2-dimensional model because we can make nice plots. Often we actually work with 3 dimensions because we found that it works a bit better, but at least here we can represent more visually the data. Now, every black point is a snapshot from either the open or the closed conformation. And we can see that kind of the points are distributed in sort of 2 lobes, where the blue point and the red points here represent the crystal structures represented on the left-hand side of the slide. Now, the color, that's an interesting thing. So this is a heuristic. It's called the dope score. It's a heuristic in Modeler. It's one of the software we use for homology modeling. It's a heuristic that estimates the energy of the protein. It's fast. So it's quite useful for us to just get a general idea on whether we're producing something that at least looks like a protein. And what we're seeing is that, unsurprisingly, the dope score is better in the regions around where the data projects. Now, the open and the closed states, they land in different regions, but they seem to be adjacent. And at this point, what we ask the neural network to do is, can you produce a movie or an interpolation between the open and the closed states, which essentially means, can we draw a straight line in this latent space and see how that interpolation looks like? Where somewhere along this line, we will have a transitional state shown in purple. At this point, you'll tell me, yeah, but it looks like your open and your closed states in the simulations are completely overlapping. So you're looking for a transition state that is already in your training set. So this is doesn't seem very exciting. But actually, the interesting thing is that this is the internal representation of the data from the neural network, but in reality, there's no interconversion in simulation between the open and the closed states. And just to convince you of that, so this plot here shows in spherical coordinates, the relative position of the little domain at the top with respect of the long domain at the bottom. So we can just basically place in space in terms of an elevation and azimuth. And we can see 2 point clouds, one for the closed simulation, one for the open simulation, and they do not interconvert. Now, that straight line I have drawn in the previous graph represents these movements you can see here. So that's 2 point of views of the transition. And that's associated with the trace made with squares on the plot on the right-hand side. And the interesting thing is that a straight line in latent space does not forcefully map onto a straight line in the Cartesian space. So in this case, you're seeing that the conformational change predicted by the neural network say that essentially you first go in one direction, then you go in another. And just for comparison, if you did this with a principal components analysis, you would predict the trace of circles. So that was quite exciting, but how do we know that what we're producing makes any sense at all? So we know it in two ways. First thing, the closed simulation is a simulation where we have the substrate, and the substrate is required to keep the protein closed. If you remove manually the substrate, the protein opens up. And it opens up like that. So that's the extra points I'm adding now. And you see essentially that they are just under the prediction of the neural network. And there's also some experimental data we had. So I have not mentioned that there are actually two structures of trapped intermediates that are present in the protein data bank. And they are the stars. So the stars are essentially in the region that is sampled by the molecular dynamics simulation here. So, and on the left-hand side of the slide, you see an overlay between one of the conformations produced by the neural network and one of the trapped intermediates we found in the protein data bank. We have an RMSD of 1.28, which makes us very happy. You'll notice that there's a couple of loops that seem to be in the wrong position. But besides that, the relative position of the domain is basically bang on, which made us very happy. Now, you have heard me talking about dop score. You have heard me talking about root mean square deviation, and those are the typical quantities we look at to tell whether the model is good. However, and this takes me to the work with Ryan. So Ryan is a PhD student I co-supervise with Antonio May up in Edinburgh, and Gregor, who is a visiting PhD student who came from Graz to spend 3 months with us. One thing that became clear is that, and that's probably a message to everyone, is not because you have low root mean square deviation, it's not because you have low energy that the stereochemistry of your model is respected. Now, we know that nature proteins are mostly left-handed, so we have L amino acids, but if it's actually possible to get a protein that contains a mixture of L and D amino acids, so left and right-handed, and still having low RMSD and low dop score, which is something not surprising. In fact, even the most recent AlphaFold model, AlphaFold 3, features about 4% of amino acids in the right-handed conformation, the D amino acids. So we had a look at that ourselves to see whether we can produce the correct stereochemistry, and the answer is, without the physics term in our loss function, the answer is not really. So this is a plot I produced this morning. So I produced lies. So it's a plot I asked my student to send to me, and I received it this morning. So here it is. So what you see in this slide is a latent space colored as a function of how many chirality violations we have. And what you're seeing is that without the physics loss function, we get, on average, every model we produce from the test set contains 4.82% chirality violations, which is in line with what's in AlphaFold. But ideally, what we would like is, no violation. And why do we want no violations? Because if we think ahead, it would be nice to have a neural network we can use to seed molecular dynamics simulation to oversample the rare events. For instance, the rare event we have seen in the movie from the previous slide of Nerdy changing from closed and open state. So the interesting thing is that if we bring in the physics term, now the region in white shows areas on the latent space where we don't have any chirality violation as well. This is quite nice. So physics penalty works. We actually did try to bring in extra penalty terms to penalize models that feature chirality violation, but surprisingly, this did not help us much at all. What helped us is literally tuning the relative importance of physics with respect of the mean square error in our loss function. Right, so now this is telling us the loss function we're using is helpful also for stereochemistry. So, right, good stereochemistry, good RMSD, good dope score. Is this enough now? And this is the last thing I would like to mention, and that's something that has been the work of Matteo, master student in my group. What we also want to be able to capture on top of all those things is the diversity of the data. So a good model should be able to reproduce the whole data distribution it's been trained with. And you can think about if you have a protein that does not really wobble much, has kind of local fluctuations, a way to produce good dope score and have low RMSD is to always return the average structure. But this is not very useful. What we want to have is the variety of structures. So one other thing we did with Matteo, to assess whether we capture the spread of our data, we calculate the pairwise root mean square deviation in our training set. Then we send the whole training set through the neural network, and we recalculate the pairwise distribution after it goes through the neural network. So neural networks tend to iron out outliers, so there is a tendency of making the data less diverse. But the question is, how less diverse is it? So this is an example where things work quite well. That's actually is MERD in 2 dimensions. And of course, we want to see a good relationship between the data, the pairwise distribution of the data in before and after it travels through the neural network in both the training and the validation set. Just to give you an example of how data looks when it's bad, this is when we train a neural network has a one-dimensional latent space. So this is not enough to capture the variety of the data. So we see everything starts clumping up. And in particular, you notice that you've got some points that lay on the horizontal axis, right? So this is especially bad. That means that actually points that are different are becoming essentially the same point after they travel through the data set. So one of the things we do, we actually we want a model that preserves the data distribution. And this we essentially look at oh, there's a question. I'll answer later, maybe. No? I need to click to see what the rest of the question.
Training data set which comes from an enhanced sampling MD improve the predicting power of the.
I think so. So essentially, this links to a point I will lean a little bit more into later. But essentially, what we would like to do in a machine learning model is to be able to capture outliers, because the outliers are our transition states, are what dictates the rates at which we switch between states. And the problem is if you say, I want to learn the outliers. So, well, that's not what machine learning is traditionally very good at. So when I told you that we captured an interpolation between an open and a closed state, essentially I said we captured a transition state, which was a rare event. But that worked out only because the 2 states are very close to each other. The bigger the conformational change, the less information we have about those outliers, the harder it gets. So one point to make is that if I have lots of information only on the states of the protein, but we have no information on the transitions, you can have 1 billion data points. This will not really help you out much. It's more having variety of data that will help us get a better model. But I'll say a little bit more about that when we get to the end of the talk. Right, so these are the typical quantities we look at. So root square deviation, dope score, stereochemistry, and how the data is spread with respect of the initial data we were training the neural network. And a nice thing of if we actually get a model that matches all those criteria is that we have now a convolutional neural network which, as I said, is an architecture that does not need to be adapted to every protein we train with. So the question we asked is, can we do transfer learning? So suppose you have now trained the neural network on MERD. What happens if I retrain the neural network on a different protein? Does this help at all? So we took our HIV-1 capsular, and you've seen this simulation a couple of times already. And what we did is, well, we tried, first of all, to train our neural network from scratch. From scratch means that all the parameters of the neural network are initially randomized, and then by showing an example after the other, we, of course, look for the weights that optimize our loss function. On your horizontal axis, you have epochs. So one epoch is when you've seen all the data once. We show, of course, the data many times. And unsurprisingly, the more you show the data, the more the loss function drops. And here, just a little extra information, you'll see that there's a kind of green band around the green line. That's an average over 10 repeats. Now, this is if you initialize the model from scratch. If you instead take the protein, the neural network trained on MERD, so the previous protein I've shown you, and then you retrain it, we get this instead. So we start from a better point and we converge to a lower value. So a pre-trained network learns faster, which is nice. And this is also true if you reduce dramatically the number of examples down to only 100. So, and this we could replicate for other proteins. This is just one of the examples. So what that tells us is that essentially the neural network is learning something fundamental about the nature of how points in space are arranged if those points represent atoms in a protein. And this information does not need to be relearned every time. And typically you can think about, bonds are bonds, angles are angles, dihedral, Ramachandran plot. Those are things that it doesn't matter which protein you're looking at. The bonds should have a certain length, angle should have a certain opening, and the Ramachandran plot will need to have a predictable distribution. And this is typically what's learned in the first layer. So this does not need to be relearned. So is this what can we use this for besides playing around with transition states? So initially I told you that our goal was to, at the very beginning of this whole story, was to do protein docking. And I've told you that the HIV capsular is a protein that's used for protein docking. So this new neural network we have can generate a conformation of our HIV-1 capsular that looks like the bound state, having been only trained with conformation from the unbound state. Now, what we found very interesting here is that now this is again a plot of the latent space. This time we're coloring it as a function of how similar that specific region of the latent space is to the bound state. And what we notice is that the best representative of the bound state is outside of the front of everything the neural network has seen. So this is essentially local extrapolation. And here I need to put a big asterisk. So this is not the neural network discovering a new state. This simply means that if we locally extrapolate, the model does not break right away. We can actually generate something that still looks like a protein that is still meaningful by pushing out from the front of what we've trained the neural network with. Now, does it always work? This is a question I asked Lucy. So Lucy came to my group for her master's project, and when I asked this question, Lucy said, "Watch me." And so she went in the protein data bank, and she found a case she wanted to challenge the neural network with. Lymphotactin, which happens to be a fold-switching protein. So the protein has been crystallized. It's known to take two different folds depending on the experimental conditions and the biological conditions as well. And we have crystal structures of both. And so Lucy said, okay, let's see if we can predict a fold switch with the neural network. And now I'm hyping this a lot, but it so turns out that actually you absolutely can't. To be honest, it was quite interesting, and useful and very informative for us to see. So Lucy simulated 1 microsecond for each of those states, collected all this data, trained the neural network, and then asked, how do I go from one fold to the other? And well, according to the neural network, you need to have a magical tunneling of a protein through itself, which, as far as I know, should not be possible. So I'm very happy to tell you why it does that, because it's quite funny. Essentially, the neural network learns how to cheat. But the bottom line is there's a limit to what extent we can locally extrapolate before the model actually breaks down. And in this case, the two states, the two folded states, are so far one from each other that anything you will predict stands a very high chance of being broken. So we cannot just discover new state. We cannot discover conformational changes that require dramatic rearrangements unless we have some data, or unless we come up with a different way of training the network. Which takes me to what we're trying to do now, and that also links us to the question we had before. So essentially, the reason why this neural network fails is because we have no information at all about anything that kind of leads one fold to become the other fold. And the question is, perhaps, can we do this online? Can we generate conformations that are with a neural network that's just outside of the front of what the neural network is seeing, run a small MD of that, and then retrain the network to force the data set to go to link our two different folds. And this is what we are developing now. And for that, we need interesting data sets to play with. This is one of them, different acting. Another data set we're playing with, and this is actually work from my PhD student, Asal. So we're trying to see whether we can play only with experimental data. So no molecular dynamic simulation at all. So Asal has fished out all the structures of calmodulin in the protein data bank. So this is calmodulin. It's a very wobbly protein. So if we overlay all the structures, what we get is two domains that really dramatically flay around. And so we have now the first models that are coming out from these data sets Asal has produced. Now, the goal of this is to see whether just from experimental data we can predict what kind of dynamics we should expect when we change the experimental condition, for instance, when we add calcium or when we add another binding partner. But yeah, thinking more fundamentally about how do we know that our models are right, the problem is that there's a fundamental lack of ground truth. So we are trying to claim we discovered transition states, but how do we know we are right? In the case of MERD, that's actually one of the very rare cases where we know what the ground truth should be. One, because we can easily obtain it with simulation, but also because we have crystal structures of the intermediates. But there's literally no other case in the protein data bank, to the best of my knowledge, that has this feature of saying we have two states, but we also know how they look halfway through. So, yes?
Some transporters are effective. Leucine family transporters.
Oh, good. So, yeah, so is that a monomer or a multimer?
Some of them are monomers, some of them are trimers.
Cool. So we actually have found recently that we can do multimers as well. So that's just a way to say, by the way, we can do multimers. So now to have an information on whether we can capture the whole dynamic conformational changes between two states, molecular dynamics are perhaps still the best way to go. But the problem is that, well, I just made the point at the beginning that molecular dynamic simulation can help us sampling the stable states, but the probability of observing a transition is inversely proportional now to the height of the barrier. So if we want to see, yes.
Sorry, do you want two-state proteins or multi-state, or what are you more interested in?
So the examples I've shown up to now are just two states. So we have an example, a protein called SIR-A, that is multi-state. It's a 3-domain proteins, and those 3 domains take many different conformations. That opens an interesting question on how much information on the conformational space of the protein can we truly capture with just a 2-dimensional lattice space.
Yeah, I mean, I was going to second the comment that made ABC transporters that have hundreds of structures in the PDB with 2 states. And then if you want a multi-state, I would go GCRs because they naturally have multiple intermediate signaling states that are also resolved in the PDB.
Yes, that's absolutely true. And there's a whole database of GPCRs and simulations as well, right? I think one of the things that at the moment has, I wouldn't say stopped us, but made us, do we really want to dive into this? It's one thing we actually want to do is to be able to seed MD simulations from conformers generated from the neural network. And as soon as you bring in a membrane as well, that's creating an automated protocol becomes trickier. So soluble proteins are just an easier solution at the moment. But yes, so if you think about soluble proteins, one thing that is interesting is, well, there are now efforts of producing really, really long molecular dynamics simulation. I'm thinking about the trajectories that the show has shared, and I'm thinking about the folding atom network as well. And that actually is something that we're looking at with Ryan and Tony up in Edinburgh. So one of the things we're looking at, and this is unpublished, we're just starting working on that. We trained our neural network with molecular dynamics of signaling, which is the smallest protein. It's a protein that switches between a folded and unfolded state. And at the moment, the interesting result we have is that the neural network separates in different regions of the latent space the folded and the unfolded state. So, of course, here we're talking about a conformational change between the folded and the unfolded state that leads to models that in between are not physical. But it's already good to see that the neural network spontaneously can separate out the two different states. And the other thing we're doing, and that's a work that's besides involving Tony and Ryan, also involved Sucrit at Sloan Kettering. So through Sucrit, we got access to very, very long simulations of several kinases run through the folding atom network. That's an example of a kinase. And so we have several millisecond or half a millisecond long simulations. And what we're doing, and this is Ryan's and Sucrit's work, we can think that if a simulation is really, really, really long, for all intents and purposes, we can treat this as a good approximation of a ground truth. If simulation is really long, it will have sampled all the transition states as well. So what we're doing with Ryan and Sucrit, we're running what are among the biggest macro state modeling ever produced. So this is just an example of them for an ABLE. And the idea here, we're thinking, well, if we have a true thorough sampling of the whole conformational state, and we want to see if the neural network is able to discover transitions between states, or later, perhaps, is able to discover new states, what we can do is to take this model and then do selective ablation. Because, okay, how about I want to study the transition between states 7 and 5, find all the intermediates, remove them, and see if we can discover them. How about I want to see if I can discover an intermediate state, for instance, state 4. Let's remove it completely from the data set and see if we can find it. Yes.
Can I just ask like a practical question? Like how big is that 1.8 microsecond simulation, and what code was used? I mean, do they use their own? I forget.
Oh, so this was there's been a combination of the deep time and PyEMA. So if the question is, are the simulations long enough?
The trajectory, sorry, I mean, how large is that?
No, we're talking about several terabytes.
Okay. And what code was used?
So these ones, they were made. They dropped all the data onto an Amazon web server, and we just pulled them from there. So that we are writing up the paper at the moment.
Desmond or?
No, that's on the folding atom network. So basically, they have a lot of very short simulations, and then they get collated to create a meta big trajectory. So that's what you're seeing here is the aggregated time. Is not a single long trajectory. From that, you can build then a Markov state model. But yeah, so the idea here is that we would like to make the models available as well. So if people are interested in doing this kind of work, you have a data set where we have actually indexed all the trajectories, say these are the different states, these are the transition states. So that would be a way for us to tell whether we are discovering something that gets us closer as we possibly can with today's capabilities to a ground truth. I think to mention, actually, are those simulations long enough? That opens another kind of worms. So we're literally interested in proteins that are having loops switching conformations, and this just scrapes through what seems to be enough. Right. So now just to close, one of the things we found is that building those models and testing them and trying different loss functions and handling different data sets, that was a bit of a pain. So we actually, if you're interested, we put together a Python package that you can, it's on CondaForge. So to do everything I described, you can just do that with between 5 and 15 lines of Python code. And the reason why I'm actually putting OpenMM in this slide is because this is the kind of, the unsung hero of this whole work, as Sam, my PhD student who just defended, spent about 6 months of his life figuring out how to have PyTorch data being passed directly to OpenMM on the GPU so that we can do this as fast as we possibly can. And so this is implemented in Molearn, which is our Python package. So if you want to do this kind of modeling it's out there. And there's even a little Jupyter notebook with an interface if you want to just to play around. Click on the latest page, generates a representation on NGL view that you can just download. I just wanted to flag that because Sam spent so much time working on that, and this is super useful also for our new students. They come in the lab, the first thing we ask them to do is to train some neural network and then display that on this interface. So it gives them a bit of a feeling of what the neural network is doing. And that's my last thing. So thank you to the people who did the work, and also so you see here two photos. The top is Durham, and I'm super grateful to Durham because that's where I started my career, and until November, that's where I was. And now I'm in Edinburgh, and I am just as grateful because they give me the chance of really pushing my research. I'm now in between informatics and chemistry. And thank you for your attention.
You mentioned that you usually have 3 dimensions in your latent space. Did you not find it helps by growing the dimensions further? Like sometimes with our methods, we use 8 dimensions or something like that.
Yes, so we did check that. And in fact, I'll try to share a slide, which will cause a little bit of chaos. But if you give me a second, I'll be able to do that, hopefully. So we did try to increase the dimensionality of our system. And now let me go back to here and share it. Right. So, so if you remember, I was mentioning that one of the things we want to capture is the distribution of our data. We want to capture the variability of the data. And the quantities we look at is the residual. So we want the data to be as close as possible to the line, and not only as close as possible to the line, but we want the data to also be to have a high cross-correlation coefficient. So we want a good spread, and we don't want this spread to be under the line or above the line. We want it to be on the line. And what you're seeing in these plots, that's an example from MRD. We get there similar plots for all the different proteins we work with. In blue is the validation set, in red is the training set, and of course we want them to follow the same trend. What you're seeing, we want to minimize the residual and maximize the cross-correlation coefficient. So notice we have a negative value here. And what you see is that you reach a plateau. The plateau, depending on the protein, is between 3 and 5 dimensions. Increasing the dimension any further does not really help much. And in fact, if you crank it up even further, you actually start degrading the performance because of the cursor dimensionality. So yeah, for us, so MRD works well enough also with 2 dimensions only. That's why it's a nice example to display. But otherwise, 3 to 5 dimension is what we find most useful.
Any other question? Otherwise, I have a question. First of all, beautiful talk. Thanks a lot. And I had a question regarding the latent space, but I'll pass it for later. So at a certain point, you were mentioning actually the effort that you are putting into using a combination with enhanced sampling. Obviously, the idea that your if you want how much the latent space is capturing, the information is dependent on your data. Have you identified ways in which you can, before you start the training, understand if you need more data, more simulations, if you have a strategy, if you need to go and ask to get some of the not close border or close front border?
To be honest, we have not done that at all. The only thing we've done as a way of processing the data that we found helps is, and that which links actually to a point that was made before on enhanced sampling. More than having a lot of data, it's better to have a data that truly captures the variability of all the different conformations the proteins can take. So one of the things we found is actually the models perform slightly better if we downsample the data, making sure that we keep the outliers, but we slightly downsample the examples of stable states. So we have a couple of different clustering algorithms that are implemented in Molearn. In general, that's something that helps. Now, we can't really tell whether the data we have is enough. So typically that only comes a posteriori. The thing I have not mentioned is how long it takes to train the model though. So on MRD, we're talking about 1,200 examples, I believe. For 200 epochs, we train in half an hour, more or less. On on a NVIDIA GeForce 380 Ti. So something you don't need a supercomputer for this. You just need a good graphic card. As long as you have about 32 gigs RAM in your computer, you can run that.
Brilliant. Any other question? I come with a question on the latent space.
Right.
So clearly, it's interesting the fact that you are able to have a latent space of only 2 dimensions, and that is really quite effective for probably everything that has 2 states and is simple enough. The question is that how much you can retrieve in terms of information from thermodynamical ensembles from the latent space? Do you see that the latent space is capturing some of that information?
Right. So I think this is a super important point, and the answer is we're essentially not caring about thermodynamics at all, which is per se not very useful. What we are thinking is that there's 2 ways you can go by with this problem. Either you try to build a model that captures inherently thermodynamics, and we've seen this, for instance, with the Boltzmann generators. That's the kind of work. Or we can try to build a model that represents the data, and then we can try to use that as a seed point to then retrieve thermodynamics a posteriori. And in my opinion, there's advantages and disadvantages for both. One of the difficulties of models that try to capture the thermodynamics right away is the scaling with the number of atoms you're dealing with is not very good. Actually, we've seen, I have to say, there's a shout out to this fantastic paper from Frank Noe that just came out on the archive, I think last week, that really pushes the envelope of what you can do in generating ensembles of conformers. But let's say in our case, we can literally train on a pore-forming toxin containing hundreds of thousands of atoms, and this works. So the way I see it is probably if you're dealing with a system that features really large-scale conformational changes or features a lot of atoms, perhaps a good idea is to go first to a model like this one, and then use it to identify essentially transition states or putative transition paths, and then restart simulation from there and retrieve the thermodynamics from there. But otherwise, for systems that perhaps have a slightly more limited dynamics or have less atoms, I mean, work with, among others, Frank Noe and collaborator, that is really exciting.
There any question maybe online to check if there's otherwise? If we have some copy or something downstairs.
Right.
Okay, so thanks a lot again to both of our speakers. Fantastic talks. Thanks a lot to everyone for